Hi, my name is Vishesh Kumar, and I am a junior studying math and physics at Arizona State University. I do research in Pressé lab run by Dr. Steve Pressé. We are a computational lab that tackles problems in biophysics using rigorous statistical learning methods. I love the research that we do because it combines programming, statistics, and applied math to tell a story that is rooted in physics. Today, I'm going to talk about my current research project in which we attempt to map diffusion coefficients across a cell membrane. The general idea is to develop a Bayesian nonparametric tool that learns varying diffusion coefficients across a cellular membrane of a cell from multiple single particle tracking trajectories. In layman's terms, we develop an algorithm that takes in the trajectories of proteins and spits out a continuous map which describes the rate of diffusion as a function of space. Let's first give some background about the thing we are tracking, membrane proteins. Membrane proteins are proteins that lie on the surface of a cell. They can move strictly laterally along the cell membrane. By tracking a protein over time, we obtain a protein trajectory. Because protein movement is diffusive, the trajectories are mostly random. However, certain properties of the trajectory are predictable, like the average frame-to-frame jump size, which you can think of as a speed. This frame-to-frame jump size, or diffusion speed, is related to what we call the diffusion coefficient. Now, if the cell membrane was completely flat and uniform, we would expect the diffusion coefficient to be the same everywhere. But as can be seen here in this turf microscope image of a cell membrane, the membrane is not uniform. And the illustration here depicts the many structures on and near the cell membrane, such as lipid rafts, the actin cortex, and lipid shells, showing in fact how heterogeneous it is. This results in membrane proteins moving faster in some areas of the cell and slower in others. This influence, caused by the structure of the cell, is precisely what we are trying to learn: which parts of the cell induce faster trajectories and which ones slow proteins down. Thus, understanding and identifying regions with higher and lower diffusion coefficients can help determine various structural properties, such as protein-lipid composition, molecular crowding, and overall cellular heterogeneity. Let's say we have approximately a 30 micrometer by 30 micrometer slice of a cell membrane where we observe particle trajectories. The trajectory data would look something like this. The zoom-in shows how each green blob in the whole data is an individual protein trajectory just zoomed out. We must now define exactly what the data actually is. Mathematically, we describe the process of proteins moving around as a 2D Brownian random walk. Essentially, the next position of the particle trajectory is randomly distributed around the current position, according to a Gaussian distribution. This probability distribution has a parameter, the diffusion coefficient, that determines the possible spread. A large diffusion coefficient means particles in that area will tend to move or diffuse more, and particles in areas of small diffusion coefficients will diffuse less. Since the diffusion coefficient is directly correlated with how much proteins move, it seems straightforward to determine its value in areas where we track the particle. But what about areas where we do not observe protein diffusion? The plot here exhibits what real-world data looks like. And as you can see, the trajectories are very localized, and this leads to many gaps in between different trajectories. This is where statistical learning comes into play. Time to jump into how we learn the diffusion coefficient. Let's continue working with the same data shown earlier. Since we want to learn the diffusion coefficient everywhere on this 30 micrometer square membrane, we need to establish a relationship between areas with data and areas without data. Recall, we mentioned that the diffusion coefficient is very well defined in areas with data, the green. Speaking qualitatively, we can say that the diffusion coefficient at any one point should be closely related to the nearest data and loosely related to data that is further away. If we take a point here, we expect the diffusion coefficient to be very similar to the data here and less similar to data here. Mathematically, this means placing a spatial covariance on the map. We now have turned discrete diffusion coefficients into a correlated continuous map over the whole membrane. Now comes another issue. There are an infinite number of points on this membrane, which means the current setup is not suited for computers. In fact, the map would be impossible to learn on an infinite number of points. To remedy this problem, we lay a uniform grid of points across the membrane and call these the inducing points. Then we learn the diffusion coefficient at these points. We then use the diffusion coefficient evaluated at the inducing points to interpolate the diffusion coefficient everywhere else. To actually do this learning, we must first define a probability distribution to determine how likely each combination of diffusion coefficients is. This distribution is found using Bayes' rule and placing a Gaussian process prior on the diffusion coefficient map. Then we use a sampling method called Markov chain Monte Carlo to explore the probability space and find the most probable set of diffusion coefficients for the inducing points. To keep this presentation approachable, I will avoid going into the math behind that. But basically, after running our Markov chain, we can use the most probable value of diffusion coefficients at the inducing points to evaluate the diffusion coefficient at every other point. Now, I would like to go over some results generated by this algorithm to verify it works as expected. We use simulated data to verify our algorithm, which means that we can compare our results to the ground truth. Here we see our ground truth map on the left and our results on the right. Side by side, we can also see that the learn map not only looks similar, but it also has a smooth continuous map, implying that the spatial covariance applied played its role. Looking at the error here, we see our learn map is within 5% of accuracy through most of the membrane, and it only begins to deviate at the edges that are far from most of the data. This serves as a proof of concept for the algorithm as it has given reliable results on simulated data. The next steps for the project is to take this algorithm and apply it to real-world data and see how the results hold up. To do this, we will first have to develop a noise model. The goal of this is to learn and minimize the influence of measurement noise. Then the algorithm will be ready to properly analyze real data and produce diffusion coefficient maps for real cells. Thank you for watching my talk. I hope you have learned something new or gained a new perspective on mathematical techniques in biophysics. I would like to thank Dr. Steve Pressé and the ASU Physics Department for providing me with the opportunity to pursue research at a high level. I would also like to thank Shep Bryan from Pressé Lab for opening his schedule to support me through this project.